This conference will now be recorded.
Great. Well, good morning, good afternoon, everyone, and or good evening. My name is Andrew Tyler. I'm the co-lead of Working Group 5 on the GEOAquaWatch initiative, which is focused around education and capacity building. And Steve Greb is the overall director of the program. Welcome to the third of a three-part series of talks featuring projects from around the community, from around the world. So I say the third, unfortunately, we have to reorganize the second one due to technical challenges, and that will be launched next week on the 19th of May. So please do come along to that. Today's theme will focus on the inland waters, focus for Earth observation. And for those of you unfamiliar with the GEOAquaWatch initiative, we are the water quality initiative within the GEO, with the goal of building the global capacity and use of Earth observation-derived water quality information to support water resources and management and decision-making. This work is mainly a volunteer effort and is supported by the many dedicated water quality professionals from around the world. So if you're interested in participating in any of our activities or working groups, we'd really welcome your talents. So please visit our website, geoaquawatch.org, or contact Mary Beth, who's also on the call and helping to manage this meeting. Her contact will appear in the chat. Before I introduce today's moderators, I'd also ask you to please mute your microphones, turn off your video, and place your questions in the chat box for the discussion at the end. During this series, we are also trying to make an effort to involve more of our early career professionals and students. So to that end, I would like to introduce you to today's moderators, and they are, to start with, Megan Kofer, who's a research fellow at the US EPA, Environmental Protection Agency, and a PhD student at North Carolina State University. And Mortimer Werter, also a PhD student at the University of Stirling in the UK. So with that, I'd like to now turn it over to Megan to take us through the first set of presentations. So thank you very much, Megan.
Yeah, thank you, Andrew. So first we'll have Daniel Odermatt from EOWAG in Switzerland with a talk titled In Situ to In Orbit, Optical Properties of Swiss Lakes.
And Daniel, do put on your microphone, your video as well. That's helpful. Thanks.
Welcome to a short presentation on how we are linking in situ and in orbit measured optical properties of Swiss lakes. My name is Daniel Odermatt. The work that I'm about to show was performed in collaboration with Gami Minodo, Abulfazl Irani Rahagi, and Johnny Wüst. We work at the Swiss Federal Institute of Aquatic Science and Technology, EOWAG, and at the Swiss Federal Institute of Technology in Lausanne, or in short, EPFL. The best data we have is acquired in this location here, which is next to the northern shore of Lake Geneva, about 400 meters from the land. In the top right of the photo, you can see a 10 by 10 meter research platform on which a large variety of limnological measurements are performed regularly. We have a concession to keep the platform there between 2019 and 2027 and hope, of course, to extend this concession. In the center bottom of the photo, you can see the TETIS profiler, which is almost 2 meters high and performs automated measurements of optical parameters. It was first deployed in October 2018, and since then it has operated about half the time, the other half of the time being needed for repair and maintenance. The TETIS profiler ascends every 3 to 6 hours and acquires measurements during these ascents, which supplied us with more than 1,300 AOP and IOP profiles up to now, covering all seasons apart from the months of July to September. Some of the data that we acquired in these periods are shown here. In the top, we have absorption line height indicating phytoplankton abundance. In the bottom, we have two plots of backscattering at 700 nanometers, indicating particle abundance and turbidity. The cases on the left represent late November and early December 2018. On the right, we see a contrasting case from the spring bloom in 2020, namely the second half of April. Besides the main parameters, you can also see indicated in these plots the euphotic depth as a white line and the thermocline depth as a black dashed line. Visualizing the data in such a manner gives us a lot of ideas of what's happening in the water and how the dynamics in lake optics come into place. One example is here on the 8th of December. We have in an otherwise relatively constant, homogeneously limited productivity, a sudden break and an increase in the thermocline to less than a third of the previous situation. This is directly related to wind mixing and a distribution of the phytoplankton that was previously concentrated in the surface layer and afterwards takes some longer time to reestablish. Very striking, of course, is the near surface vertical gradient in the spring case, where the thermocline depth is considerably lower or considerably shallower than the euphotic depth, leading to these deep chlorophyll maxima, which will significantly affect the relationship between absorption and other optical parameters and the water-leaving reflectance. Contrasting, we have a very nicely mixed surface layer in the less productive part of the year. We also see, for example, an intrusion down here where we have a highly turbid water that reaches the Lexplore platform at the depth of almost 40 meters. This intrusion is associated with the Rhone River, which plunges into the lake at several dozens of kilometers from the location of the Lexplore platform. But from there, the turbid water is transported to the platform at this depth without a lot of mixing. So this particle freight is out of reach for any remote sensing sensor. And it's very important to understand such processes also when interpreting or making conclusions from remote sensely data. Apart from these insights in lake processes based on inherent optical properties, we can also perform standard operations using the TETIS measurements of apparent optical properties, which usually means a comparison of atmospherically corrected reflectance and ground measured reflectance in the first place. With the TETIS measurements, we can additionally simulate and model a second ground-based in situ reference reflectance using the absorption, attenuation, chlorophyll, and backscattering measurements from the TETIS profilers. In this way, we obtain an uncertainty estimation by comparing the two ground measurement-based reflectances, flag the measurements that achieve a better match from the two sources, and then use only the quality checked and better matching reflectance measurements for validation. In addition, we also included an optimization step because we found that the backscattering measurements performed by the sensors on TETIS are often not very sensitive, especially during clear water phases. So we included this optimization step, which not only allowed us to get better and more realistic reflectance out of the simulations, but it also revealed a nice seasonality in the backscattering, which is shown on this figure. For the entire period in which we have TETIS data available, even though the samples may come from different years between 2018 and 2021. On the x-axis, we see scattering at 550 nanometers measured by the sensors on TETIS, and then we have the optimized backscattering at 550 nanometers. Both are fairly low towards the turn of the year or at around the turn of the year. But then in springtime, as productivity increases, we have a drop in backscattering on one hand and continuous increasing in scattering on the other hand. The backscattering ratio finally increases again after the end of the productive season, when the nutrients around the middle of the year are depleted in the surface layer. And accordingly, we have a larger impact of inorganic particles again. By the way, the dashed line in black represents the Petzold phase functions backscattering ratio. So you can see that this is around the seasonal average for Lake Geneva. The data measured at the Lexplore platform and by the TETIS profiler is increasingly made available in a portal called Data Lakes, which is accessible at GEOAquaWatch-datalakes.ch. In this portal, we do not only want to make accessible the field sampled measurements from automated stations as well as our campaigns, but we also want to combine it increasingly with hydrodynamic simulations, as indicated by the surface currents with these white lines, and remote sensing products as this chlorophyll map, which is in the background represented by blue and green colors. In such a visualization, you can immediately see how the two are related, and by providing this information along with the in situ measurements, we also want to inspire users to take new looks into additional data sources and promote both hydrodynamic simulations and remote sensing in this manner. Our remote sensing products are produced with this Python package called Zencast, which is also publicly available. It essentially wraps a number of different snap operators, tools such as Polymer, and even terrestrial algorithms such as Cent2Core. It downloads Sentinel-2 and 3 data automatically from different data sources, performs data processing jobs according to specified geographic perimeters and time intervals. And with a custom configuration of the processing job, you end up with a number of different products. We are currently validating these products for different lakes in Switzerland. And while we already have some dummy remote sensing products from Sentinel-3 in data lakes, we will replace them this summer with a validated and error quantified product. We also have additional automated stations like this WISP station in Greifensee, for example. And on the left, you can see a visualization of how we present reflectance data in data lakes. So in summary, we showed how Lexplore enables unique optical measurements for aquatic science, as well as remote sensing, and especially on the interface of these two disciplines. We have high frequency dial measurements from the profiler that support standalone products. We've seen the particle and phytoplankton abundance, but we elaborate this further even to primary production estimates. We've seen examples of how it supports bio-optical modeling and Earth observation product validation. We process our data and disseminate it using a number of open access tools, which you can find under the URL specified. And we want to make increasing numbers of data sets available in this portal, and some of our future fieldwork will also target alpine lakes. And our ambition is to provide this future data, this campaign data, in the same framework and using the same processing techniques. With this, I want to close and look forward to some questions or comments. Thanks a lot for your attention.
Thank you so much, Daniel. And just as a reminder, you can put questions in the chat. Our next talk will be from Priyanka Jamwal from the Center for Environment and Development at the Ashoka Trust for Research in Ecology and the Environment in India, with a talk titled Data Transparency, Moving from Grab Sample to Real-Time Water Quality Monitoring.
Hello everyone, I'm Priyanka. I am a fellow at a research institution called ATRI. It's a private research institution based out of Bangalore, India. So today I will be taking an example of Chakkur Lake in Bangalore. I will be talking about the drawbacks or the problems with the conventional way of water quality monitoring and how Earth observation can help us in addressing those problems. So, giving you a little bit context of the problem, India is facing huge water quality crisis, and one of the reasons for this water quality crisis is that 90% of our wastewater is discharged untreated into our surface water bodies. Now, if you look at the city of Bangalore, and if you look at the graph on your left, you will see that this population of Bangalore city has doubled in last decade, whereas the infrastructure growth has not kept pace with, especially the wastewater treatment infrastructure has not kept pace with the increased demand for wastewater treatment, and as a result, we see untreated wastewater getting discharged into our surface water bodies. Because of this, there is a huge loss in the benefits which are provided by these surface water bodies. We have seen lakes on fire. We have seen foaming and frothing in our lakes, and also there has been in water bodies which have thick layer of chlorophyll A and harmful algal blooms on their surface. Now, again, taking an example of Bangalore city, I will be discussing the drawbacks with the conventional methods of monitoring water quality. So this is the map of Bangalore city. It is around 857 square kilometer with 12 million people. You can see here that Bangalore city is located in 3 valleys, and each of the valleys has a network of lakes. Now, again, the point here to note is that these are not natural lakes, but the man-made lakes which historically were used for irrigation. Currently, these lakes provide multiple benefits such as groundwater recharge, environmental amenities, irrigation water, fisheries, and biodiversity. In spite of providing all these benefits to the local stakeholders, still these lakes face various issues in terms of reported frequent fish kill events, algal bloom, frequent bird deaths events reported across lakes in Bangalore. Now, one of the reasons for the poor management of these lakes is because there is a lack of understanding on the processes which govern water quality and water quantity of these lakes. Now, the reasons for the knowledge gap is that the key variables are very, very dynamic. Because the catchment of these lakes are urbanizing, the quantity and quality of inflows keep varying, like they are very, very variable. So developing mathematical models for such water bodies and to managing them, it's very difficult, extremely difficult. And then there are trade-offs between the functions under different management regimes. Also, very little is known about on how to manage lakes to serve these multiple functions required from them. Now, at H3, we tried to understand the processes which govern the water quality in the lakes, and for that, we wanted to understand the quality of inflows, the in-lake water quality, and develop some kind of a database which can help citizens, NGOs, and even government stakeholders to manage the lake better. So we started with the conventional way of monitoring lakes using grab samples. We collected data. We collected samples from the inlet, from the outlet, and from within the lakes, and monitored for various water quality samples. Now, this is Chakkur Lake. Its area is 64 hectares, and it has these interventions, which is like, and there's a sewage treatment plant to ensure that treated effluent enters into the lake, and it also has constructed wetlands to ensure that nutrients are scrubbed and removed before water into the lakes. So we collected water samples at the inflow, outflow, and at the center of the lake to understand the dynamics, the changes in the water quality with respect to the inflow inflow water quality. Now, again, you can see here that the dissolved oxygen levels that levels during the middle of the day and during the afternoon are very, very high, like, they are double of the saturation value, whereas the levels which are required for the propagation of fisheries are just 4 milligrams per liter, which means that the quality of lake is good enough for propagation of fisheries. And this data also confirmed with the data provided by regulatory agency, which says that it falls in category D, which means that water quality is good enough for the propagation of fisheries. Now, in spite of all this data coming from our end, which is a private research institution, and coming from the agency side, says that, like, the water quality is good enough. But in spite of all this data, we were seeing frequent fish kill events at the lakes, which was definitely hampering with the management of lake water quality, and citizens were unable to spin their head around the kind of data we were providing and actually what was happening in the lake. So we tried to change our water quality quality graph sample regime to understand the temporal and spatial variability in dissolved oxygen. And here you can see a graph on your left that water quality dissolved oxygen variability across the depth of the lake. And here you can see that early morning dissolved oxygen levels are below 4 milligrams per liter, which tells you why fish has been dying in the lakes, because fish is under stress as the levels early morning dissolved oxygen levels are very, very low. Again, we looked at the spatial variability of dissolved oxygen levels, and we found that the upstream and downstream dissolved oxygen levels were very, very different. And we also looked at the chlorophyll A levels, which gave us an idea that almost the lake is always in hyper-eutrophic state, which means that the chlorophyll A levels in the lakes are very, very high, which is leading to this very high dissolved oxygen variability, which further leads to frequent fish kill events in the lake. Now, to address these issues with the graph samples, we thought of, okay, let us upgrade and use ResearchGate standards with IoT to create this dashboard where citizens can be provided with this data, real-time data, and then better actions can be taken, or better decisions can be taken to manage water quality of lakes. So this is how we envisioned a citizen science dashboard for lake, where there is a problem, and then we have these sensors who are monitoring and providing us real-time data on the water quality of the lake, which is informing and empowering citizens, pushing government to take action and to address these problems. But in spite of using all these sensors, when we applied them on the field, what we found was that after a few days, the sensors required frequent cleaning because there were fouling issues. And so we really struggled, and then we said that, okay, it was, rather than becoming a real-time monitoring, it was just like, some person has to visit the sensor daily to clean the fouling. So it was not helping us with monitoring at the lake in real time. So then we shifted to citizen science mode, where we trained the volunteers who were interested in environmental issues to monitor water quality. Now, the first thing was to get these low-cost sensors, which are kind of accurate. So we collaborated with the startups who were developing these sensors. We helped them calibrate their sensors in the lab, in the water quality lab at E3. And once the sensors were calibrated, citizens were trained, and after that, the data was used in the web application by using open source code. We were using this data to upgrade our lake dashboard, and then the data was available for stakeholders, for citizens to take actions. And so these are a few pictures from the site where you can see that for managing water quality and using Internet of Things and real-time sensors, it's a very, very, very tedious task. But when we use citizen science, we were able to provide data in the context. So the way we created these lake dashboards were providing data in context where citizens were able to make sense of the data. Like, for example, if nitrates are high, are above 10 milligrams per liter, citizens don't know what to do with the single point data unless and until you provide context of why this particular parameter is important. So that is how the dashboard was created. And so here we argue that to further strengthen our dashboard, the lake dashboard, it is very, very important to integrate Earth observation to monitor water quality, because we can definitely use citizen science to create these algorithms to calibrate the Earth observation data, which are very, 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 very apt for the local conditions, for the tropical conditions, and further then it will help us in improving data transparency, empowering citizens, will help with informed decision making, assess the impact of interventions on water quality, and increase accountability. Thank you.
Megan, you may be on mute.
I was not on mute. My internet was just struggling for a second. Sorry about that. Thank you, Priyanka, for your presentation. Our last one before the break is going to be by Leif Olmanson from the University of Minnesota in the United States, with a talk titled "Advanced Remote Sensing Methods for Automated Lake Water Quality Monitoring," or Water Quality Mapping. Sorry.
Let me give you an update on the work we've been doing using satellite remote sensing for water quality assessments in Minnesota, and specifically how we've automated the image processing procedures with the collaboration of David Porter at the Minnesota Supercomputing Institute. In Minnesota, we have a good field monitoring program for water quality variables. At the same time, we have satellites regularly collecting imagery. So what we do is we find some good clear imagery, link up the field measurements within a few days of the image, build a statistical model of that relationship, apply that model to all the lakes on an image. Do that for enough images, we come up with the water quality for the whole state. Using those procedures, we've completed 9 statewide assessments since 1975, with over 10,000 lakes included in each of these assessments. These are at around 5-year intervals. We use that for analysis of spatial and temporal trends and causative factors. One thing we did back in 2002 is we made all of our data available on an online Google Earth format lake browser, where people can zoom in and get information about their lakes. It's been very popular with thousands of unique visitors come to get their information every month. Having water clarity at 5-year intervals has been great. It's been real popular, but if we really want to improve lake and fisheries management, we need more water quality variables more often. And that's where we're going with these new sensors that are available. They have higher spatial, spectral, radiometric, and temporal resolution. Not only can we map water clarity, we can also measure the factors that control water clarity, things like chlorophyll, suspended materials, and color dissolved organic matter. The spectral characteristics of the sensor determine how well it can measure different water quality variables. And for these 2 satellites, Landsat 8 and Sentinel-2, Sentinel has additional red edge bands that are very useful for chlorophyll measurements, picking up this peak here. And so for Landsat, works really well for water clarity and for CDOM, and for Sentinel has a little bit better for the chlorophyll. Another big advantage of the Sentinel-2 satellite is the high temporal coverage, which I'll demonstrate here with some imagery from August of 2019. As you can also notice, there's lots of cloud cover in Minnesota, and so we're going to need a lot of coverage in order to cover the whole state really well. Most of these images I would not use for my manual techniques, so automation really helps. And so basically having this imagery, an opportunity to build a near real-time water quality monitoring system, and that's what we've done here. We have all this new data available, downloaded to the ground stations. We can have a machine-to-machine access, pull it into the Minnesota Supercomputing Institute. Once there, we can kind of do the hard work here is in this box right here is basically preparing the images for the applying our models. So things like atmospheric correction, which is basically just removing the effects from the atmosphere from the imagery to what would be expected. Found lots of different atmospheric corrections. We've been testing a lot with the A6 paper that just came out, and then also with a lot of stuff we've been doing in Minnesota. They're all give different results, so they're not all the same, and some are better than others. And we found that the main atmospheric correction works best in Minnesota and for inland waters. So the other thing is cloud masking, and this we basically decided to just use the cloud mask 4, but we add an extra buffer. There's some artifacts left over with that, so we have to buffer it, and then we have additional masks on top of that to clean up all the artifacts for inland waters. So with that, we were able to get some pretty good results, but we started having these anomalies. And in Minnesota, we've been getting a lot of wildfire smoke from Canada and from California. And so those were kind of affecting it. If you look at here, looks like the wildfire smoke is picked up in the terrestrial areas, but not over the lakes. So that can be real problematic for what we're trying to do. So we had to come up with another aquatic smoke mask. And so that's what we did, and we were able to take care of that. So once we have just clear pixels left, we can make some water quality models. So for that, what we did is we basically linked up all of our field measurements to all of our satellite data, and then we found the ones within, for this case, 1 day within of an image. And for the second disk, we're at over 6,000 measurements for just in 2019. For the other years, we have over 20,000 measurements. And the model developed in 2019 fits the data very well. So we have very good models. Similar thing with chlorophyll with over 10,000 measurements there. So we have some water quality models. We can make some maps and some tabular data, distribute it to different groups and put in our lake browser. So that's the stage we're at now. Hopefully by the time you see this, we'll have it in our lake browser. You can look at it there or in our temporary lake viewer that I showed you. An example of the number of data points we're able to get using this methodology. This is an example from Sentinel-2, May through October. You see, we do have good statewide coverage. There is more imagery in the overlap area, so where the blue and green dots are, than there is in the gap areas. What we have found is that we're able to do pretty much monthly mosaics on a pretty consistent basis. There will be a few gaps, but for the most part, it works out really well. And so that's our approach. We've done monthly pixel and then statewide lake level data as well. And all this data is available on our temporary website or on the lake browser that was in the URL earlier. Let's show you some examples of the water quality data that is available. So we have chlorophyll, clarity, and CDOM for every lake in the state on a monthly basis. And here's the International Rainy River, Lake of the Woods watershed up on the Canadian border, Minnesota Canadian border. And you can see a fairly pristine area. So there's not a lot going on for water quality, but this is 2019. I'm just going to go show you some examples. There's this one little red dot here that shows up for all these little lakes. There is a lot of activity, a lot of algal blooms in Lake of the Woods, which is well known, and then upper red. But for the most part, this is a pretty pristine area, just lakes and forest wetlands. And so that one dot that showed up, we took a little closer look and the imagery, and we found, yep, there was a bloom that day. So here's an example in a little bit more dynamic area. This is the Twin City metropolitan area, and then the surrounding agricultural areas around it. And this is 2019. So May, June, July, August, September, and then October. And then we will go to 2020. Very similar patterns for both of these time steps. July, August, September, lots of blooms. And very interesting. We can dig in a little deeper and do this at the lake level too. So this is at the lake level for daily time steps. So here's Lake Francis, more of a mesotrophic lake. Titicaca, which is a more eutrophic lake. Washington, Cicada, and German are all eutrophic, and Elysian, that is actually a hypertrophic lake. So all this data is available. There is, if you look at this gray one here, that is actually not in the overlap area, whereas the others are in the overlap area. So you can see the difference in coverage. So still good coverage, even if it's not in the overlap area. Okay, here's another one, more of the monthly time steps. Gives you a nice graph showing the dynamic of what's going on in these lakes, and very interesting phenology for the algal blooms. We've also been having good success in other areas, neighboring states, South Dakota and Wisconsin, and then some of the southern United States like Georgia and Alabama, which is a lot more clear imagery down there, and a little bit more dynamic systems with the reservoirs due to more storm events that come in and show good pulses, which is kind of interesting to see. And then some other thing in Lake of the Ozarks in Missouri, when you see the pulses come in from storm events. Enhanced lake browser coming soon. So look forward to taking a look at that. If you get a chance, URL is down here at the bottom. With that, I have some acknowledgements, and that is about it. So enjoy the conference, and look forward to any questions you have.
Thank you so much, Leif. We'll now take a 5-minute break, and we'll reconvene at about 40 past the hour for the next set of presentations.
Yes, and I think we are back from a short break. And I am Mortimer. I'm introducing the next two speakers, and we'll also lead a bit the Q&A session and discussion. And we continue with Petra Philipson from Brockman Geomatics in Sweden, who is going to give a talk about cyano alert.
Welcome to this follow-up presentation about cyano alert. My name is Petra Philipson and I present this on behalf of the cyano alert team and my co-authors Carol, Kerstin, Irini and Susanne. The present cyano alert service is the result of technical developments and validation efforts performed within the Horizon 2020 project cyano alert. And that ended in June 2020. When the project was presented at a GEOAquaWatch webinar 2.5 years ago, the work was in a planning stage with very few developments implemented and tested. The project partners are shown here, and this talk is a presentation of the achieved project developments and where we are today, almost 1 year after the project has ended. cyano alert is now an operational service for organizations concerned by health risks and quality of water resources, which need information for monitoring and management of lakes and coastal areas. The service is now provided by two of the implementation partners, Brockman Geomatics and Brockman Consult, and one of the project user partners is now a paying customer. The targeted goals for the initial project was 1, to develop a warning and monitoring service based on Copernicus data. 2, to develop a service platform and a fully automated production process. 3, to customize it to the user requirements with help from the involved user partners. And 4, to reach a high level of integration or adaptation to the customer's workflows. The technical developments have been focused on the service tools, a viewer, alerts, and an app, and all with strong emphasis on the actual end user need. The viewer allows for near real-time display of data and products and a time series analysis by the user. The alerts correspond to information messages with contents shaped by the user and sent to the user on the same day as the satellite overpass. The app is open and free and provides latest estimates for the selected location and allows for the user to report their observations in the field. And before I show some examples of these tools, we will take a look at the service system behind this. This is an overview of the developed system environment, and in this sketch, we take off from level 2 products in the upper left corner. For each service region, a level 2 data cube is created, which means that the level 2 products are selected with respect to the needs and requirements of the user, that they are fit for purpose with respect to the water type, cut, filtered, and formatted and stacked in a cube, which is then updated near real time when new data is collected. To do this, we use Xcube, which is a Python package developed and maintained by Brockman Consult, and it's used for generating and exploiting data cubes. The cubes and related tools is then the basis for the viewer, for the alerts, and for the app, which I will show you examples of in a minute. In the earlier slide, we started from level 2 data, but if we now go backwards, we also need to produce the level 2s. And we start from Mary's Sentinel-3 and Sentinel-2 level 1 data. During the project and also continued afterwards, all hardware needs was assessed and secured, and processing chains for each sensor, combining all necessary processing steps, were defined and implemented. The output from the processing chains is then selected, formatted, and added to the level 2 user cube. The developed service is called cyano alert, but also includes products corresponding to other water quality parameters, the normal suspects, like turbidity and TSM and Secchi depth, for example. All are listed here, And we have also implemented a chlorophyll product corresponding to a merged version of MPH and C2RCC chlorophyll products, which presently is also being upgraded for improved in performance. We will now leave the data and move on to the tools, and we start with the viewer. The viewer is the web app to view and look at multidimensional data stored in a cube generated with Xcube. It is easy to use also for a non-expert user and gives near real-time access to the data stored for the specific user in the user's cube. In this example, we are looking at Lake Mälaren, west of Stockholm in Sweden. And this is the viewer, and what you see here is the RGB, the corresponding chlorophyll map, and also the cyanobacteria indicator map. So the viewer allows to look at the data and the products, but it also has functionality to add user-defined points and polygons and to generate time series on the fly for these points and polygons. In this example, I have drawn and plotted time series for 2 different areas in western Mälaren. And in addition, the user can, for example, select between mean or median plots and to add or to not show error bars. The second part of the service corresponds to an information subscription service. And based on predefined locations and criteria, near real-time information is extracted from the user's cube, summarized in a report, and sent to the user via email, if this is the method that the user prefers. In this example, subregions of the Baltic Sea were used as a basis for an alert service. Different alert risk levels were defined based on chlorophyll A concentration, and the result was plotted in a map, summarized in a report per subregion, and sent to the user. In this case, the full criteria for issuing a high risk for algal bloom was that at least 20% of the area should correspond to a chlorophyll concentration above 4 microgram per liter, and this was decided and set by the user. The example subregion is called North Proper Baltic, and the report also include percentage of pixels, which concentration was over the alert level, as well as percentage of valid pixels after cloud screening. The third part that I would like to mention today is then the CyanAlert app. The app is based on the Cyan app, which was developed by the US EPA and then adopted to CyanAlert needs. It is presently covering the Baltic Sea drainage basins, but it grows with the new users and user areas. It's available on Google Drive since 2020. The data available in the app corresponds to the 2 latest chlorophyll A estimates for a selected location, and also information regarding risk for cyanobacteria presence. The left image shows the start window, and in the right window we have zoomed in and tapped in a lake. The report then shows the chlorophyll A concentration, if this is less or more than the earlier estimate, and if the cyanobacteria flag is activated. In this window, it's also possible to prepare and send a report. If one click on report this location, a new window pops up and a text and photos can be included and submitted. It is also possible to select a color on the water and see example photos if you click on the information button to the right. After submission, the reported color and location becomes visible to other users of the app. Finally, not all users are only interested in near real-time information, but many organizations and authorities are rather interested in seasonal summaries or annual reports in specific formats. So different templates and reporting structures have been elaborated to meet the customer's wishes. And this example shows a report in the shape of a summary fact sheet produced for one of the German customers with information from Sentinel-2, Sentinel-3, and in situ data. And with this, I would like to say thank you for listening, and I look forward to questions and for the discussion later.
Thank you very much, Petra, for your presentation about CyanoAlert, and we go on to our last presentation from Peter Hunter, who's also working at the University of Stirling, and is going to give us a presentation about the UK Lakes Observatory and the operational water quality products from Sentinel-2 MSI data.
Hello, and thank you to everyone for joining this webinar. I am Peter Hunter from the University of Stirling, where I'm a senior lecturer in Earth observation. In this talk, I will provide an overview of the UK Lakes Observatory, a new pilot service developed for algal bloom monitoring in the UK. The funding for this research largely came from the UK Natural Environment Research Council. I must also acknowledge the wider project team involved in this work, not only my colleagues at Stirling, but also collaborators at the UK Centre for Ecology and Hydrology, the University of Glasgow, and our SME partners, 3DO. It is also important to emphasise that this work is by no means standalone. Rather, it builds on the considerable achievements of the NERC Global Lakes project, which also involved significant contributions from the Plymouth Marine Laboratory and the Universities of Dundee and Reading. The two main drivers of cyanobacterial blooms in the UK are nutrients and climate. Previous work by our project team has shown that lakes in the UK are likely to be vulnerable to climate-driven increases in blooms, not only through the effect of elevated water temperatures on growth rates and nutrient release, but also as a result of decreases in river flows and flushing during summer droughts. In this study, published in Global Change Biology, we showed that lakes in the UK, particularly those north of 55 degrees latitude, are particularly vulnerable to such changes. However, our understanding of these impacts and the risks they pose to water quality and human health is significantly hampered by the fact that monitoring is currently infrequent and often reactive. The overarching objective of this project was to examine whether satellite data could be used to enhance bloom monitoring and underpin a new climate service for the UK. As part of the aforementioned NERC Global Lakes project, we developed methods for remote sensing of water quality at global scales using ocean colour satellites. But these methods are not directly applicable to the UK because the vast majority of our water bodies cannot be reliably observed at the 300 metre spatial resolution afforded by these ocean colour satellites. Therefore, in this project, we had to look at the higher spatial resolution data provided by Sentinel-2. Sentinel-2 provides multispectral data at a native resolution of between 10 and 60 metres, which is much better suited to the scale of water bodies we find in the UK. So through the funding we received from the UK Climate Resilience Programme, we built a new pilot digital platform, which we have named the UK Lakes Observatory. This is the overview of the processing chain that underpins the UK Lakes Observatory. I don't think I have time to linger on the technical detail, apart from to highlight the fact that the processing chain employs an ensemble of algorithms for the estimation of chlorophyll, which are dynamically mapped to the type of lake under observation. This allows the processor to adapt better to the spatial and temporal variability we see in the optical properties of lakes. This short video shows some of the functionality of the UK Lakes Observatory. The initial map shows the location of all lakes for which we process data, grouped initially by region. Currently, we are processing data for just over 900 lakes. This initial map layer allows users to toggle lakes on and off based upon their chlorophyll status and trend. This allows a user to isolate lakes already displaying bloom events, or to show those with recent deteriorations in water quality. The user can then zoom slightly to reveal a map layer showing the current water quality status for all the lakes monitored at the UK scale. Here, the symbology shows the chlorophyll A concentration as well as the short-term trend. We can see, for instance, that there's some lakes in the Norfolk Broads that are currently showing quite high chlorophyll A concentrations. The user can then scroll or search for a lake of interest. Here I'm searching for Alton Water, a small drinking water reservoir near Ipswich in the south of England. We can then zoom to that water body, and at this scale, we're now presented with a new dashboard showing some basic information about the lake, which we ingest from the UK Centre for Ecology and Hydrology's UK Lakes portal. At this scale, we can now view the satellite chlorophyll time series for this particular water body. And we can select any observations within that time series and display the corresponding chlorophyll product from Sentinel-2 on our map view. The data are currently produced weekly, although we can process the data daily if necessary. We also have the provision to issue real-time alerts to users should a lake or set of lakes exceed a given threshold concentration. The algorithms implemented in the processing chain behind the UK Lakes Observatory were trained and validated using available in situ monitoring data provided by the Environment Agency for England and the Scottish Environmental Protection Agency. Here we see that the estimation of chlorophyll in eutrophic lakes affected by blooms is pretty reasonable, particularly during the so-called bloom season, which typically runs from midsummer to early autumn. We currently struggle, however, to estimate chlorophyll accurately in other low productivity lakes, but this is something we're working on behind the scenes at present through other projects. The comparisons are perhaps more interesting and informative when you compare the data at the lake level. Here's an example from Esthwaite Water in the Lake District, which is a eutrophic lake with a history of algal blooms. The monthly chlorophyll measurements from the Environment Agency are shown in red, and the Sentinel-2 chlorophyll data are shown in blue. Underneath, bloom events are identified in both time series based upon the exceedance of the 80th percentile value estimated independently from the two time series. You can see that, although we underestimate compared to the monitoring data, the satellite observations do a reasonable job of tracking the seasonal and interannual variation in chlorophyll. We also capture the majority of bloom events identified in the in situ monitoring data were within a window of plus or minus 1 week. The UK Lakes Observatory is very much in its infancy, and we are continuing to work on all aspects of the processing chain. In addition, one of the next phases of this research will be to better integrate the satellite data with other in situ monitoring networks, such as the citizen science data provided by the UK Centre for Ecology and Hydrology's Blooming Algae smartphone app. We are also working to integrate the data into a new smart catchment observatory called Fourth Era, alongside data from IoT sensors and models based on artificial intelligence. Finally, Just to summarise, what we've demonstrated through the development of the pilot UK Lakes Observatory is that firstly, Earth observation can be used successfully to widen the surveillance of algal blooms and increase the likelihood of early detection. We see this approach as being very complementary to conventional and other novel monitoring approaches. It is not a replacement for lake sampling, but it can be used to greatly enhance the quantity and quality of the data we collect. Ultimately, when employed operationally, the data provided by Earth observation has the potential to significantly improve our understanding of climate impacts on water quality in the UK and elsewhere. And I will leave you with this animation showing all the bloom events captured by the UK Lakes Observatory over the lifetime of the Sentinel-2 mission. Thank you once again for listening to this talk, and I very much look forward to the panel discussions to follow.
Thank you very much. And this was our last presentation for today. Mary, would you like to address some words, or shall I start with the Q&A?
Good afternoon, ladies and gentlemen. My name is Evangelos Villosopoulos, and in my capacity as the director of the Greek Geo Office, one of the 3 colleagues of the Urban Heritage.
There was a video you had running in the background.
Oh, I'm sorry. I'm trying to get that to turn off. So I think you can just go ahead and start the Q&A. I apologize.
Great. No problem. We have a question from Nimisha, who has a question for our second speaker, Priyanka Yamwal, about her presentation. And she is interested to know how once the data that is, when it is made available, how it drives, how it could drive a change in policies.
Thank you, Namisha. It's really very interesting question. And so let me answer your question by taking example of Bangalore. So as you know, you might know that Bangalore has around 120 lakes. And out of 120 lakes, around 45 lakes are restored. When I say restored, it means that there is some kind of intervention done. Either there is a sewage treatment plant or constructed wetland to ensure that treated effluent enters into those lakes. Now, if you look at the policies which are there with respect to restoration, the restoration is like, the policy says that lakes, the restored lakes should be able to meet either drinking water standards or they should be good for swimming, right? Now, these are the policies which just comes out of the blue, because if you look at the kind of influent which is entering into these restored lakes, which is secondary treated effluent, it is very, very difficult to achieve these standards unless and until a huge investments are made in terms of treatment. Okay, so when you have, if we have data for these restored lakes based on their catchment characteristics, based on the kind of interventions and the quality which we have in these restored lakes, we can provide an evidence that given this amount of investment and given this level of intervention, this is what is achievable and this is what we can attain in Bangalore. And then we can formulate policies with respect to those evidence, because at present, if you look at those 45 lakes, none of the restored lakes are compliant with the policy. They are not compliant because the goals we have set are unattainable. Like, we can't have lakes where we have inflows coming in which are secondary treated water, but we want to achieve drinking water standards. So that is why what my argument is, that if we have these kind of evidence to show linkages of catchment characteristics, interventions, and lake quality, and across several lakes, then show that with these kind of inputs, this is what is attainable, then we can have better policies, and where, most of the lakes will able to comply. Thank you. I hope I answered your question. Thank you.
Thank you very much, Priyanka. Nimisha, any follow-up questions, or is that all right?
Sorry. Thank you, ma'am. Thank you for that answer. I was very intrigued by when you mentioned citizen science, because I worked on a college project using citizen science for climate change, for light pollution, actually. But this was very interesting for me to know that we can sort of do a lake surveillance using citizen science, and it has the potential to drive change, and it's not just an optimistic point of view. So thank you, and thank you to the other speakers as well. It was a great, insightful session. I look forward to hearing all the question answers. Thank you.
Thank you, Ramisha. Thank you.
Thanks. And our second question is from Philippe Lisboa, I think, that is directed towards Leif's presentation, who is asking about how good is the Landsat 8 model for estimating chlorophyll A concentrations.
So our approach has been to use Landsat for the water clarity and for CDOM, and to use the Sentinel-2 for the chlorophyll, CDOM, and the water clarity measurements. With the big change with the collection 2 from Eros for the Landsat product, it's been very difficult to get the imagery out of there, and so we're still working on our Landsat. We have the Sentinel-2 operational, but the Landsat is we're still working on it, and but I do not expect we'll be using it for chlorophyll, but we will definitely look at if there is a possibility of using it for that. The big thing is the temporal part of it, and that Sentinel-2 has a lot more temporal coverage than the Landsat.
Right. Thank you very much. And we have a question from Stephen Grapp towards you, Peter, who's asking which lakes were demonstrating a decline in water quality and what kind of metrics methods were used for this analysis, and how many observations over what time period and so on.
Yeah, thanks for your question, Steve. So the trend indicator that we estimate at the moment is based on short-term changes in the chlorophyll concentration. So let me get this right. What we do is we look back over a 6-week period, and we try and identify, I think it's a minimum of 3 observations in that period. And if we see a consistent decline or increase in the chlorophyll A concentration over that period, that's how we get to a trend. So it's just sort of an indicator of what we think might be happening in that particular lake at any given time. The idea is that it sort of accompanies the bloom indicators. So you've got, on one hand, you've got a way of identifying which lakes are perhaps subject to blooms at the present time, but also those lakes where perhaps there's been a declining water quality, you've seen an increase in chlorophyll A, and therefore it might be the case that a bloom will develop in the coming weeks or indeed months. What I would say there as a caveat, we haven't validated that trend indicator yet as extensively we have for the chlorophyll concentration. So that's something that's still to do, but it's potentially useful because obviously water quality managers want to know potentially what's going to happen in the weeks and months to come, not just what's happening at the present time. So we thought it was a useful addition to just a simple concentration value.
Thanks, Peter.
Any follow-up, Stephen?
That was great. That's what I wanted to know is kind of what type of metrics they're using for this trend indicator. It's something we've been considering here in the Wisconsin lakes.
Excellent. Peter, you have a second question, actually. Vinny Raman would like to know whether you have a lake-specific chlorophyll algorithm, or do you use a common algorithm based on all observations from all lakes?
Okay, yeah, thank you, Vinny, for the question. It's not a straightforward answer. We actually have more than one, effectively we have more than one version of the processing chain, and the different versions use a slightly different approach to how we map algorithms to water bodies. But in essence, what we do is we have a number of optical water types, which you might be familiar with. So we perform a classification in the first instance to identify the optical water type. And there we have two different versions. We have a version that uses a collection of 13 or actually slightly more optical water types, which was based upon a publication by Evangelos Varakos and colleagues. And then we also have a simplified version of what we've termed optical water classes. So the first step is to identify those optical water types or classes, and then that determines what algorithm is used for that particular water body. So effectively, we have a little suite of algorithms that are trained to different types of water bodies, from say high chlorophyll to low chlorophyll. As you've probably seen from the presentation, we do a much better job, as you might expect, at the high chlorophyll end of the scale than we do at the bottom at the present time, which is fine for monitoring blooms, but obviously for maybe other applications where you're interested in what's going on in those low productivity lakes as well, we need to effect some improvements there.
Thank you, Peter. And there is actually a question from Lawrence towards Peter, yourself, and Leif, who would like to know if GEOAquaWatch can help you, and also the platform and the network can help you learn from each other and develop the services to other parts of the globe, especially lower income regions. And maybe we start with Petra this time. Otherwise, please just fill in if you would like to give an answer on this question.
Yes, not a very easy question to answer, but I mean, just by arranging these seminars, we get an update on what everyone is doing. We've been collaborating with Sterling before, and I guess you're still collaborating with my colleagues in Germany on many things. And we share the pool of algorithms and so on, but presently not a strong collaboration base at the moment. I don't know how to formulate what you can do to help us.
I can maybe come in and add to that a little bit. I think, as Petra says, we have collaborated in the past, probably more so on things like algorithm development than on the actual operational services, but probably the latter is something we could collaborate on more going forward. So maybe that's something that AquaWatch could help facilitate, perhaps through some working groups or workshops or something like that. I think what is particularly interesting is how we communicate information to our users and what our different experiences are there, because that's obviously something that we've been working on as part of the project behind the UK Lakes Observatory, is how we provide the information in a form that's useful to a range of different stakeholders. So at the moment, our service is being evaluated by both environmental regulators like the Scottish Environmental Protection Agency, nature conservation agencies like NatureScot, and also water utility companies, but they all have slightly different needs and probably slightly different requirements in terms of how they would want to access the data and what sort of, say, indicators are provided. Some won't be interested in images at all, they'll want numbers, others might well be interested in some of the spatial information there. So I think that type of information and experience could be usefully shared across projects where there's been a focus on developing operational services. And we might even need to work towards, let's say, some common standards there as well, so that that information is provided in a let's say, a slightly more systematic and digestible form across different services.
Yeah, in Minnesota, most of our funding has come from the state, so we're kind of focused on Minnesota, but we're definitely interested in expanding outside of Minnesota to other areas. Now that we have our system operating, for the most part, there's always, improving the methodology as we go, and that takes research dollars to do it. But I think the biggest thing would be finding funding would be a great thing for if you really want something, you got to fund it. So it does take time and resources to do it.
All right. Thanks everyone. Oh, sorry, Lawrence. Go ahead.
Yeah, I was just going to say sorry about the difficult question, but you know, I think I'm thinking it from a GeoAquaWatch perspective, whether there's anything we can do to particularly support you, and whether it is, as you say, having workshops, getting you together to discuss operational difficulties or challenges and solutions and things. I think that might be something we could follow up. But if you do think of anything in terms of lessons learned or you know, improving knowledge sharing between sort of operational systems, I think that would be something that GeoAquaWatch would be keen to pursue and support somehow.
Just to come back in there, even maybe something just as simple as putting some information on the website might be useful as well. It's possible that there already is some information on some of the services on the GeoAquaWatch website, but I think certainly our experience since we've sort of developed the UK Lakes Observatory is that it's been really useful in terms of just facilitating discussions with some of the stakeholders that we're working with. And it's useful to be able to put something in front of them and demonstrate what is possible in terms of operational services from Earth observation in a much more effective manner than you can achieve than just showing them a few plots or a scientific paper. It's really helped open that discussion, and I think that's an effective way forward. So maybe there's probably other organisations out there that would be interested in the range of services that have been presented today. So maybe some of that information was put on a website that people could then identify and then follow up on. That might be also useful alongside the kind of workshops they've just discussed.
Yes, we have another question actually for Life as well. Do you think there is enough existing in situ water quality information available through the EPA or other open source data sets to expand your tool to corners?
Yeah, I think there is actually. I mean, you know, previously with our, you know, we're doing our manual things would have, you know, in their later years, I mean, starting you'd have 50 to 100 measurements for validation, calibration validation. Now, then you know, later days was more like 300-400 within a day of an image. But, you know, with being able to use all the imagery, with the approach of correcting the imagery and applying a specific model, I think there definitely would be plenty of data. The work we've done in Minnesota, I mean, it works well in other states that we've tried it in. So that I think it's expendable. I think it can be extended to other areas. I think the optical water types is something we're also looking at with the dominant wavelengths and the different optical water types and seeing if there are some improvements in that approach. And we have a big data set. We can start looking at a lot of that stuff. I mean, the big thing with for monitoring in Minnesota now, it's kind of a paradigm shift. You know, this data hasn't been available at this temporal coverage, and having this kind of, you know, really kind of opening eyes at the agency folks what could be possible with it. So that's kind of what we're hoping to target next, is to, you know, how can this data be used for actual actionable science at the agencies and local level.
Right, thank you. And we have a question actually that was posted from Lawrence earlier about Daniel's presentation. Daniel already halfway gave an answer, I think, to this question, where Lawrence asked whether or not the seasonality that we saw on the backscattering plots are there mainly due to particles and water. And if that is the case, if your processing chain incorporates a correction based on Earth observation measurements for this, or is that, you know, a bit circular and you prefer to use your in situ measurements of turbidity and chlorophyll?
Yeah, I answered in the chat. I'm not sure, Lawrence, whether I satisfied your curiosity. As a matter of fact, this we noticed while doing the optical closure that this backscattering contribution seems to be the ill-defined part. So that's where we tried an optimization, but we now also started to only acquire additional data to find out yeah, what max capturing measurements we can rely on, or what estimates. But that's, yeah, kind of the finding is that it's the weakest part in our closure at the moment.
I think your answer is pretty good, actually, Daniel. But I'll follow it up if there's little bits of it I'm not quite sure, but I can follow that up later date, I think. No, looks interesting how you're doing it.
Daniel, would you like to say something about the two links that you also posted about the open source software and processing of CD2
yeah.
and 3?
I'm glad to. Thanks, Mortimer. That's actually also concerning the role of AquaWatch. I think sharing software is an important task that where we can help each other out, especially when we see that feature life and what we do is an operational processing chain for a pretty national or state focus. So we kind of clearly delineate our areas of interest, but still we can use very much the same software for a lot of generic tasks. And I think that we can save a lot of effort when we exchange software more extensively, given that all the basic, or a lot of the basic algorithms from the Snap toolbox that are used in all the processing chains are open software already. So I think we just have to make sure that we also exchange the integration tools on top of it. This, yeah, for us, it consists of the backend, this Zencast tool, but then also the data lakes portal is an open visualization framework. I'm not sure I wasn't aware of Peter's visualization tools. I think there are some comparisons to be made of what we might want to integrate from this functionality. This looked very interesting as well. But I think that's a big potential for AquaWatch.
Thank you, Danny. Maybe a question from myself as well that's related to this for the data visualization portal, but also the Cyano Alert app, and then there's the Bloomin' Algae app coming up in the UK Lakes Observatory. Are there any lessons you learned or things that did not work out as anticipated in your project, which might help others in the community to be aware of? And that's obviously a question for, well, basically all speakers who are in a project related to it.
I can maybe kick off with something. I'm not sure if this quite answers your question, Mortimer, but one thing that we have found as a challenge when we've been developing the service is that the vast majority of data that we have for calibration and validation of algorithms has come from the UK regulatory agencies. But the vast majority of those data are grab samples taken from a somewhat unknown location on the shoreline, which is far from ideal when you're trying to validate and or tune algorithms. And then that's a bit of an issue because we know there's big uncertainties potentially with those data and using them in the way that we do, that we're then sort of hardwiring into the algorithms to an extent. So there's that big spatial mismatch where the field data, the in situ data, is a grab sample from a shoreline where you might have relatively high chlorophyll A if there's been downwind accumulation of cyanobacteria or something like that, but we're estimating a chlorophyll value based on a whole lake median or mean, or potentially a kernel of pixels. The availability of really good quality data, not so much quality data, but data better suited for algorithm validation is somewhat more limited, and that's more of a challenge, I think, when we start to evaluate how good some of the algorithms are and how how effective the products are that we're producing. And it's something that I think we need to tackle. I'm really jealous of Danny's system. I'd love to have a few of those on lakes to help with validation.
Yeah, in Minnesota, I think we've been kind of spoiled with the amount of field measurements that there are available. If you look at the CONUS level, Minnesota is kind of a hot spot of field measurements, starting real early and continues to this day. Most of those are kind of directed by the state agencies, and they're in the middle of the lake typically, so it works good for remote sensing. Yeah, samples around the shoreline aren't very useful for us. I think so the big thing with Minnesota is the Citizen Lake Monitoring Program. So that was started in the 70s, and it continues today, and that's a great program, collecting both chlorophyll and also other parameters and Secchi measurements. But still, it's only about 10% of our lakes that are monitored on a regular basis, so the remote sensing really helps to fill in the rest of the 90%.
Thank you. And I think we have to, Petra, can you comment after a question we have from Priyanka, actually for Priyanka from Andrew, because Priyanka has to leave. I come back to you. I did not forget you. Changes in behavior to reduce society's impact on the environment is one of our key motivations, says Andrew. And Priyanka, could you comment on this from the work you've been leading, including the citizen science engagement? Has there been a demonstrable change in the behavior for the better of communities?
So, Andrew, I can talk a little bit about behavioral change in from not my lakes work, but another work which we did, a project which we did with the James Hutton Institute, where we deployed decentralized wastewater treatment system and intervention at the school in a rural village. There we worked with the community, and we saw demonstrable behavioral change in the way students were using water, and the quality of gray water which was coming out from those from the school. So, but coming back to the lakes, so if you look at how lake is getting contaminated, it's not a particular household or a particular person which is going directly and throwing or discharging some contaminant. So we have not seen any kind of behavioral change per se in a particular person, but as a community, what we have seen is that community has started asking questions and have become more vigilant on what kind of water is entering into their lakes. Like, for example, in Jakkur Lake, they have community itself has formulated a committee where early morning people just walk around the lake to make sure that there is no illegal discharges happening in the lake, or there is no solid waste dumping which is happening in the lake. So that way community has come together. I don't know whether you will put it under behavioral change, but that is something what we have seen, that community organically have come together to ensure that the wastewater treatment plant is functioning efficiently. Now they can't measure the quality of effluent coming from the wastewater treatment plant, but of course, just by looking at the quality of water which is coming out, they can, or the smell, they can sense whether the treatment plant is functioning efficiently or not. And if it is not functioning, then they go to a sewerage agency to make sure that everything is fixed and a treatment plant starts functioning efficiently again. So these are the kind of changes what we have seen, but at a particular, if you ask me at a particular person level, we have not seen any kind of behavioral change. But of course, at the community level, yes, community has become more aware, and they have started asking more questions and have made state more accountable to their actions. I hope I answered your question.
You did. That's excellent. Thanks, Priyanka. That
Thank
sounds.
you, Andrew.
And has the data helped empower the community as well, or?
Yes. So ATRI, as I mentioned in my talk as well, that there was a lot of questions. So when we were doing all this monitoring, initially we were just monitoring for during the daytime, like, between 9 and 5. And what we were seeing was that grab sample data was showing very high dissolved oxygen levels. But despite of high dissolved oxygen levels, there were fish kill events. So there was a lot of confusion on why this is happening. If the water has high DO levels, then why fish is dying? So then when we did extra monitoring, where we looked at sunrise, sunset, and afternoon data, and we presented that data to the community, then they understood that, okay, this is the reason, and then we need to control nutrients, because that is the nutrients which are leading to chlorophyll A. And now there are talks about upgrading constructed wetland to make sure that more nutrients are scrubbed before effluents are discharged into the lake.
That's a great example. Data empowering communities and making change. That's brilliant. Thanks, Priyanka.
Yes. Thank you.
Thank you very much. And Petra, if you want to come back, I think you had a contribution still before I cut you off a little bit about if there are any lessons learned over the project or in situ data availability and quality as well that feed into the project.
Yeah, I can only acknowledge what Peter and Leif said about the validation data. Of course, it's more difficult to convince a user or customer when you move up from your known home lake to something new, and especially as EO data is mostly used where there is no in situ data. So that's of course tricky. But in the end of the day, we meet a lot of people who say, we really need your data, and then it's funding, because they already have so little funds to spend, and it's sort of already dedicated to what they need to do within the in situ sampling. So great interest, but not so many funding possibilities for many users.
Thank you. I think that was our last contribution for today as well. Thank you, everyone, and all speakers. Mary, if you would like to close, please take the word.
Thank you very much. Congratulations to everybody. It was a great job. I'm glad that there were very few technical difficulties this week, since we've had so many in the past, and I'm hopeful that you all will join us on May 19th at 2 p.m. UTC for the rescheduled Estuaries and Transitional Waters themed project updates. You can find more on our website, and I've mentioned that in the chat.
So this is Steve. I again would like to compliment and thank everyone, our presenters and our hosts and moderators. This has been these have been wonderful presentations, and I'm really impressed. I just wanted to touch quickly on what the point that Lawrence is making about AquaWatch and maybe our role or niche in the community. And I've always tried to, use a philosophy of focus our activities on community issues, be it the open source software, validation, which is an important issue, the in situ data, all these things that we've touched on. And our role here is, of course, to try and advance the science and the applied nature of it. So if you have additional thoughts or topics, feel free to reach out to me or Mary Beth, and we can try and help support these kinds of community efforts. So thanks again. It was a great job, everyone.
Great. Thanks, everybody. Have a great day.
Thank you.
Thank
Bye.
Bye.
Bye. Bye.